Good day to everyone. Today, the Multiple System Atrophy Coalition is introducing a brand new webinar series. The title of the series will be From Bench to Bedside, the Promise and Hope of MSA Research. The series will highlight researchers who applied for and won a grant from the Coalition's Seed Grant Program. And that program, which began in 2013, has resulted in 49 MSA-focused research projects for a total of 2.5 million. So these grant proposals address any of 4 distinct disease area categories: pathogenesis, diagnostic biomarkers, preclinical, and clinical are submitted each year. Those proposals go through a very rigorous review process by MSA researchers who volunteer their time to support this work. The finalists are approved by the Coalition Scientific Advisory Board and ultimately by the MSA Coalition Board of Directors. So everyone in this process is dedicated to finding a treatment or cure for this disease. In a sense, we all have skin in the game. So let's introduce everyone who's going to be a part of the webinar today. We'll start with the researchers whose work will be the focus of today's webinar. Dr. Wassilios Meissner, apologies. Can you please share a short summary of your interest in MSA movement disorders and a little bit about yourself?
Yeah, I'm a professor of neurology at the University Hospital and University in Bordeaux. I'm running currently for more than 10 years the French Reference Center for MSA, together with the colleagues from Toulouse, another city in France, where we are currently following more than 300 patients with MSA, which has allowed us to better understand the disease, not by following the patients, but also through different research projects. And beyond the clinical research projects, I'm also doing some translational research project, meaning that we're also going back to the bench. And this is a little bit what we're talking today about. And I will share some preclinical results that may in the future become interesting also for the patients and have impact to the patients.
Great, thank you. I love that translational approach. And Dr. Erwan Bezard, could you also share a short summary of yourself and your involvement in MSA research?
Absolutely. Hello, everyone. So my name is Erwan Bezard. I am an INSERM research director. So INSERM is the equivalent of NIH in France. I work too in Bordeaux, and I work together with Professor Wassilios Meissner. I am a scientist, meaning I do not see patients. I work only with experimental models, mostly animal models, and my main interest is about translational research for Parkinson's disease and all atypical Parkinson's syndromes, including MSA research. And we've been working together with Wassilios for the past 15 years on both Parkinson's disease and atypical Parkinson's.
20.
Parkinsonism.
20. Sorry,
oh,
20.
20. We are aging too fast. And today I will, after the presentation by Wassilios, I will present some very recent results that will ideally fit with the presentation of Wassilios, how they are exactly the follow-up and the attempt on our side to further understand the molecular mechanisms behind the insulin resistance. And it sounds a bit mysterious, but you will know a bit better what it is about after the introduction by Professor Meissner.
Thank you. Very, very excited for your talks. So there were 3 members of the MSA community that were given the opportunity to put this webinar series together. Before going any further, I'll just quickly introduce myself as one of them. My academic background is in neuroscience, neurochemistry research, but I've since moved into the commercial side of science in my current role at Charles River Labs. But I lost a parent to MSA 10 years ago now, and I've had a personal interest in continued research in this area. So I'm really honored to be a part of this coalition and to help further awareness in events like these. So that's a little bit about me. Ariana Pitaro also helped put this webinar together, and she may be joining us later on. But also Larry Kellerman, who's online. Could you give a quick introduction?
Yes. Hi, everyone. This is Larry Kellerman, and I am a former caregiver of an MSA cerebellar patient and also previous caregiver representative for the coalition. And I just can't get away from this work. I love watching the researchers and listening to them and seeing how we are going to beat the beast, so to speak. And before we go on, we do want to read one line from the slide you have on your screen. This is an educational session and it is not intended for the purpose of providing medical advice. So hopefully you guys realize that as we go through, and any questions you have, you might put in the question and answer session or section we have. Thank you, Karina.
Oh yes, thank you for that, Larry. So the two grants we're going to focus on today fall in the category of more preclinical research. The first one was submitted in 2015 by Dr. Wassilios Meissner, entitled Glucagon-like peptide 1 agonists for treating MSA, a preclinical proof of concept study. The second one we'll focus on was submitted in 2016 by Dr. Erwan Bezard, titled GRK2 targeted knockdown as a therapy for multiple system atrophy. Both focused on a problem based on observations in lab studies, a link between insulin resistance and MSA. As they're going to note during their presentation, these studies have helped move forward our understanding of this phenomenon and has led to further research and even a compound now being tested in a clinical trial. So the first research to share with you today is Dr. Wassilios Meissner. Dr. Meissner, thank you so much for taking the time to share with the audience today. And with that, I will pass it off to you. Yes.
Thank you very much. It's absolutely my pleasure. And I would like to start with saying that we are so grateful to the MSA Coalition and to all the patients, to the entire community who are making possible our research and have helped us to move forward. And hopefully we will make the lives of our patients in the near future much better. So I will present this very complicated title. I have simplified the title so that you may better understand what we're talking about. We're talking about insulin resistance and exenatide. This is an antidiabetic drug that has been used for many years for diabetes. And this is a drug we have tested in one of the preclinical mouse models of MSA. But of course, the question is, what is insulin resistance? And I just move forward to the first one, just to provide some background and introduction. Of course, many of you will know that glucose is uptaken by the gastrointestinal tract, is then traveling to the blood and will then stimulate receptors in the pancreas, and the pancreas will then release insulin, and insulin will then make sure that the glucose in the blood will go to different organs, in particular the liver, the muscle, and the fat tissue. And when it comes down a little bit to what happens exactly at the level of the cells, this is illustrated here on the right side. So increasing doses of glucose will then stimulate the pancreas. This will then lead to release of insulin, and insulin will bind to receptors which are on the surface of the cells, and binding of insulin to the receptor will then stimulate inside the cells glucose transporters to move to the plasma membrane, as illustrated here, and that will then allow the cell to take up the glucose and to eliminate it from the blood, and to enable the cells to use it to create energy, for example, and muscle action. What happens, or what do we understand by insulin resistance? And this is a phenomenon coming from the diabetes field. First of all, of course, insulin resistant means that when you stimulate this receptor, or that this stimulation will no longer imply or will no longer lead to the same reaction or lead with the same strength, moving these glucose transporters to the plasma membrane. So in other words, the body cells of the liver, the muscle, and the fat will respond less well to insulin. And then in the next step, this is what occurs in diabetes, then the concentration of glucose will increase because glucose is no longer cleared from the blood. So this is what is happening in the liver, in the muscle, and the fat cells, and this is how the body, our human body, is using glucose. But finally, we have learned over the last 20, 30, 40 years that these receptors are also expressed on many cells in the brain, and beyond function, the involvement in glucose transport and energy metabolism, they are involved in many other functions. So this is an extremely busy slide here that shows you that by the stimulation of these insulin receptors in the brain leads to many activations around these cascades. And these different cascades have not only the function to make available glucose to the cells and to help with energy balance, but also to support and protect the neurons and induce and ensure oligodendrogenesis. So what is this about? Oligodendrocytes are the cells which are strongly involved in the disease in MSA, because in these cells accumulates a protein called alpha-synuclein. So that makes this a very, very appealing target to look if there are changes of insulin's resistance in neurons and in oligodendrocytes in MSA. And this is exactly the question we asked, that was highly inspired by work in other neurodegenerative disorders, especially in the Alzheimer's field, where we know for many years that there is impaired insulin resistance in the brain, and that insulin resistance contributes to the neurodegenerative process. And we were wondering if this observation is somehow a broader mechanism that could also be found in other disorders such as MSA and also Parkinson's disease, because in our research we are also interested in Parkinson's disease. And that was the starting point. And from there then we had the chance to access to brain tissue donated by patients and to look if in these cells there were features of insulin resistance compared to a control group. And this is what is shown here. You can see, so the marker is a little bit complicated. The marker is called IRS 312. But what is important here is to see the intensity of the colors. So this is a brain of a control subject, and this is a brain of a patient with MSA. And as you can see here, the signal of insulin resistance is much stronger in the patient with MSA. Of course, the question here is what cell type, what is the underlying cell type? And this is why we did another staining for a specific marker of neurons. And when you do the overlay, you can very clearly see that this comes, so this signal here is a signal that comes from a neuron. And again, when you look at the control group, you can see that the intensity of this insulin resistance marker is lower than in patients with MSA. And when you go then through all the different subjects we have analyzed and the many cells we have counted, you see that in the group of MSA patients, there is increased insulin resistance in this particular brain area, which is a brain area called the putamen, which is strongly involved in the process, in the disease process, whereas an important amount of cell loss. We were then looking at another second marker of on a marker on the same marker in another cell type, the famous oligodendrocytes. These are the cells where the alpha-synuclein accumulates, and as you can see here again, so we did the same approach. The staining for the insulin resistance marker, and then the staining for the marker for this specific cell type. And here you can see the overlay that there is a relevant number of cells showing insulin resistance. And then again, if you look at the intensity of all the cells we have analyzed, you can see that in the group of MSA patients, there is a increase of this insulin resistance marker, meaning that in a specific region, in a specific brain region where there's really strong, the process, the neurodegeneration is really strong, there is significant insulin resistance in MSA brains. We have also looked at, within this study, in other areas where there's less neurodegeneration, especially in the visual cortex. And interestingly, in that visual cortex, we couldn't see differences between controls and MSA patients, meaning that this is probably a feature that is really in line with the cell loss we can see in specific regions in our patients. We were then wondering to what extent, because I mentioned already the alpha-synuclein, and this is here one illustration. This is one oligodendrocyte from a patient with MSA, and you can see here in red the alpha-synuclein that accumulates within this oligodendrocytes. And we were of course wondering to what extent the insulin resistance we observed in the previous slide could be connected to alpha-synuclein inclusions. And this is why we did again staining for the insulin resistance marker, but also for alpha-synuclein as illustrated here. So this is alpha-synuclein in green. Again, the marker for insulin resistance in red, and you can see here that there is a close signal between insulin resistance and alpha-synuclein, but there's no overlap. So this is an oligonucleotide, and you can see that this oligonucleotide, where is this signal, this alpha-synuclein, has high intensity, a higher signal than the other cells around. And here again, we have counted an important number of cells, and we found that indeed cells where you can see these deposits of alpha-synuclein show a higher amount of insulin resistance than the other cells. So we don't really understand currently what the meaning is, but there is a clear interaction between alpha-synuclein and insulin resistance. Coming from there, we're wondering if one of the mouse models we have been using in the past for 15, 20 years, if in that mouse model, we would also be able to find similar changes of insulin resistance in the areas that are highly involved in the disease process. And if this is the case, if treatment with an antidiabetic, and here is then when exenatide pops in, if treatment with this antidiabetic would have beneficial effects on the progression, on the cell loss, and also on the insulin resistance that may be observed in this mouse model. And the answer is yes, as illustrated here. Just two illustrations that summarize a little bit the findings in the mice. As you can see here, this is the control group. This is the MSA mouse model. And again, you see there's a clear significant increase in the same insulin resistance marker we have also assessed in human brain tissue. And then in the second step, these are the results here. As you can see, we had 3 treatment groups. That was the placebo group. That was a group with a low concentration of the antidiabetic. And this is the group animals that have received higher concentrations of the antidiabetic. And what you can see here is that the high dose of the antidiabetic decreased the increased insulin resistance. And so we were able with that approach to decrease insulin resistance. But of course, the main question was then, was this enough to have a positive impact on cell survival? And here again, the answer is yes. So we looked at dopamine neurons. Dopamine neurons, there's a loss of dopamine neurons in MSA. Dopamine neurons are important to, or if there's a loss, that causes slowness of movement, stiffness, and tremors, so symptoms, classical symptoms that can be observed in MSA patients. And when we looked at our different groups, this is probably here the most important figure. As you can see here, this is the placebo group compared to wild type. There's a significant loss of dopamine neurons. And when you look then at the lower dose and the high dose, there is really significant preservation of dopamine neurons. So in other words, in our mouse model, we were able somehow to recapitulate findings of insulin resistance, and then with a treatment, a 3-month treatment with this antidiabetic, we were able in that mouse model to preserve dopamine neurons. Within the mouse model, we were then in collaboration with Dimitrios Kapelkinianis from NIH in Baltimore. He has developed a technique, or he's one of the researchers using a technique called exosomes. And exosomes are, this is a cell. Finally, all brain cells release small vesicles, and these small vesicles are called exosomes. And what is really interesting is that these exosomes are can travel from the brain to the blood, and we can detect them in the blood. And these exosomes include a variety of proteins and include also markers of insulin resistance. And we're wondering in our mouse model if in blood we could measure somehow as a window, as a mirror of what's going on in the brain, if you could measure in the blood a brain insulin resistance marker. And finally, what we found was quite interesting, that the higher the neurons, the higher the survival of neurons in the brain. So this is just the previous slide what we have seen here. The higher the survival of neurons, the lower the concentration in the vesicles, in the blood vesicles, that came from the brain. So that was a highly interesting result, and that motivated another project that was funded later in 2017 by the MSA coalition, where we looked into patient samples in patients with MSA and Parkinson's disease, and also controls, if there were differences in these different exosomal markers. Of course, how did this move forward since we published the results in 2017? So the study was funded in 2015, then published in 2017. And there is currently a small clinical trial ongoing, a treatment trial ongoing. This is work led by Professor Fultini in London. It's a single center, so one center, open label, which means that patients are receiving the treatment without or with knowing that they received the treatment. So you may be aware that when it comes to the development of drugs, usually in the more advanced phases of clinical development, patients there's a placebo arm, and neither the patients nor the doctors know what treatment was assigned to a specific patient. But usually, or sometimes in very early trials, when we just would like to see if there's a signal, if we would like to better understand potential safety issues and side effects, then there may be an open label trial. And this is what the colleagues in London have been selected. 50 patients followed for 48 weeks, and the primary endpoint is the difference in total unified MSA rating scale score, which is a scale that allows us to assess motor symptoms, also symptoms about, for example, constipation, bladder dysfunction, syncope, fainting, and that gives a score and is a classical endpoint used in clinical trials. So, and that, no surprise, in that trial they will also look at the progression of UMSS scores. But again, this is a very, very early, small, open label trial that at best will give us a signal to encourage us to go for a larger clinical trial. And at the moment, I would really strongly advise you to not seek for open label or off label treatment with antidiabetics as long as we have a clear clinical trial. And just a final conclusion slide. I've shown you that insulin resistance is present in the brains of MSA patients and also in MSA mice. The antidiabetic drug exenatide shows positive effects on insulin resistance and cell survival in these MSA mice. And a first small trial explores currently the effects of exenatide in MSA patients, quite early in clinical development, but we will see over the next few years where this approach leads. Thank you very much. And I pass over to Dr. Bezard.
Yeah. Before we go to Dr. Bezard, I do want to say we have some questions that the three of us who are helping host this event to ask. We were going to ask in between the two, but we haven't generated any questions from the audience yet. So we're going to go ahead and we'll continue. And the next speaker is Dr. Erwan Bezard, and he will share with us his study and what he has going on today as well. So Dr. Bezard, if you are ready, we are ready for you. And thank you for joining us. Are you there?
I think we lost him, but
I think we must have lost him. In the meantime, we've lost Dr. Bezard. We'll be getting him back. Must be a technical fix. But we do have our third member of our group. So Ariana, if you would like to turn on. Oh, now we have Dr. Bezard.
No, yes, sorry, I apologize. An internet connection issue. I'm sorry.
That's perfectly fine. We thank you. I was going. Ariana had just joined us, and I don't see her right now. I think we'll introduce her after you are finished, if that's okay with you. So we'll let you go right ahead and introduce your work and talk us through it. Thank you, Dr. Bezard.
Okay, thank you. Thank you, Larry. And so the topic is going to follow exactly what Professor Meissner has presented, and the title is a bit enigmatic. GRK2 targeted knockdown as a therapy for MSA. Sounds a bit strange, but you will understand easily what it is. So I am going to skip the first slides because you see they are exactly the same as Professor Meissner presented. So I'm going to focus here on the fact that we have seen 5 minutes ago that insulin receptor signaling is compromised in MSA, but is also compromised by alternate actors. So we decided to look into molecular events that result ultimately into this impairment of the insulin transmission. We look at the literature, and as you can see from this slide, there were a number of papers not dealing with brain diseases, but dealing with obesity or diabetes, hypertension, or heart failure, all conditions resulting from insulin resistance. And all of this work pointed out that a molecular marker, a particular protein called the G protein coupled receptor kinase 2, so GRK2, that's easier to pronounce and to understand, is in fact integrating several transduction pathways and is a physiologically relevant inhibitor of the insulin signaling. And it's not a recent knowledge. As you can see here, the first demonstration has been done in 2004 already. And since then, you have this number of papers, all of them not in brain conditions, but in, let's say, in peripheral conditions. What is GRK2 and what is it doing? So what we call the canonical role, meaning the classic role of GRK2, is this involved into the modulation of signaling passing through a number of receptors to neurotransmitters. So you have GRK2 being represented here. So it is basically aiming at stopping the signal that is transmitted by these classic receptors for neurotransmitter. Another non-canonical, so non-classic impact of GRK2 is represented here. So here you have the signaling behind the insulin receptor, a bit different, but it is basically exactly the same as the one presented earlier on by Professor Meissner. But when you have an overexpression, so an increased expression of GRK2, it is capable of phosphorylating the insulin receptor and therefore inhibiting the activation of the different signaling cascades. So you have seen from the presentation of Professor Meissner that MSA is characterized by phosphorylation of the insulin receptor, and that this phosphorylation is in fact a marker of resistance to the insulin signaling. So we posited that if we were capable of decreasing this expression of GRK2, so decreasing the levels of the protein GRK2, we might improve the insulin signaling function. It has been shown in the periphery, but in the brain as well. And of course, it is here clearly known that it is leading to a phosphorylation on a specific position. And Professor Meissner and myself showed in the mouse model introduced by Professor Meissner that yes, there was an increased phosphorylation of this specific residue on the insulin receptor. So we first started by investigating the brain of MSA patients, generously given through the French brain bank, and we worked on a number of control person and a number of MSA patient looking at different brain areas. So we look at the forebrain, so the striatum and the frontal cortex. We look also at the pons and the brainstem. And we found out that there is an increase, a significant increase, as you can see here, into the MSA patient, into the forebrain, not in the pons. Also, they might have a tendency, but clearly significant and relatively high expression of GRK2 into the forebrain of MSA patients. So there is an increased expression of GRK2, and if you remember the presentation by Professor Meissner, there is also an increased insulin resistance in the brain of the same patient. So what did we do? We did a test into the mouse model of MSA that we use in the lab, and we first asked the question, if we were injecting a molecular construct capable of reducing the expression of GRK2 into the striatum of this mice, would we be then capable of preventing the MSA features in this mouse model? So you have the timeline here. It's basically, you do an administration, what we call a stereotactic administration into the brain of this mice, and then we wait 24 weeks before to collect the brain. So they are, of course, investigated for their behavior. And then we have a number of endpoint molecular markers that we are investigating. And Professor Meissner has shown you, for instance, a number of dopaminergic neurons, a number of the load into synuclein and the load into phosphorylation of the insulin receptors. So we worked with two groups of mice. So PLP-SYN stands for the mouse model of MSA. So those are mice expressing synuclein into the oligodendrocytes, and they received a construct which is scrambled, so it's not supposed to do anything, and a construct that is supposed to downregulate, to decrease the expression of GRK2. When we looked at the behavior of the mice, we showed that these MSA mice were displaying less impairment than the mice having received the scramble vector, meaning they were improved. We looked at the number of neurons, so very comparable to the graph earlier shown by Professor Meissner. And again, we have a nice preservation of the number of dopamine neurons into the substantia nigra of this mice compared to the mice having received this scramble vector. The total number of cells is preserved. We looked at synuclein. While the total amount of synuclein presented by these mice was not affected, the presence of what we call high molecular weight, so HMW stands for high molecular weight species of alpha-synuclein, so aggregated form of synuclein likely to be pathologic, is in fact decreased in the MSA mice having received the vector supposedly leading to the decrease in GRK2 levels. We demonstrate also that this was accompanied by a decrease into the phosphorylation of the insulin receptor, and therefore we can say that these mice are showing lower insulin resistance. And very importantly, this is correlating with the fact that we do have a lower levels of GRK2 in the brain of these mice, meaning that yes, we have engaged a target. We did in the mice what we were supposed to do. By decreasing the amount of GRK2, we were likely capable of obtaining a better behavior preservation of the number of dopamine neurons into the nigra, a decrease into the synuclein pathology, and a decrease of the phosphorylation. So what we can say is that when you do in adult mice, when you inject into this mice molecular construct aiming at decreasing the total amount of GRK2, then you prevent the development of MSA features in this mouse model. So such an invasive approach is conceptually amenable to patients, and this has been done not necessarily in MSA, but in disease like Parkinson's disease. Gene therapy is definitely possible. However, the delivery here in this specific setting was highly localized, so we injected into the striatum, and of course in MSA, at odds with other conditions like Parkinson's, the pathology is widespread. So it's not simply a question of preserving the dopamine neurons into the nigra or preserving the neurons into the striatum, but it's really looking at an approach which would afford protection into the whole brain. So we were wondering, could brain-wide delivery achieve similar results and open the way then to truly effective strategy? So here we adopted a strategy which is slightly bizarre for most of you, but in order to target the whole brain with exactly the same molecular construct, what we do is that we inject the molecular construct, not anymore in adult, but in utero, so into the uterus of the pregnant female. So you have a representation of a pregnant female. You take outside of the belly the uterus, and we were able to inject into each embryo a small amount of the viral vector into the brain of each embryo, and then you put it back into the belly. You do a suture and you wait for the delivery of the animals. And we had shown in the past that it is a useful strategy for obtaining a transfection of the whole brain. So we were quite confident with the approach. So what we do here is exactly the same as with the adult mice. At the, of course, major difference that the administration of the viral vector is done in utero at the embryonic age of E14, so meaning 14 days after the mating, while the births occur around day 20, day 21. And then you wait, you do the behavioral investigation, and of course you terminate the animals in order to look at the readouts that I have presented earlier. So we have 3 groups this time. We use wild type animals, so classic animals, normal animals, and they have received a sham surgery while being in utero, and the 2 groups of PLP synuclein mice, so the MSA mouse model, receiving either a scramble molecular construct that is supposed not to do anything and actually does nothing, and the molecular construct that is supposed to decrease the availability of the GRK2. So the question was, therefore, is in utero delivery of this molecular construct capable of preventing MSA feature in this mouse model? So in mice developing MSA over time. So what you can see here, this is a group of interest here, the MSA mice having received this molecular construct aiming at decreasing the amount of GRK2, and we do have an improvement of the behavior. In fact, it's relatively comparable to the normal mice, similar age, that are still performing better, but they are not statistically significantly different from these MSA mice having received the GRK2 construct. The number of neurons is also preserved, a bit lower than into the wild type, but clearly there is a significant improvement compared to the PLP mice having received this scramble molecular construct. The high molecular weight species, so I remind you the let's say the load into the pathological synuclein, if you want, the aggregated form of synuclein, it was significantly decreased into this mice having received the molecular construct aiming at decreasing the load of GRK2 in a way, in a magnitude very much comparable to the wild type animals, so to the normal animals having not received anything and not being a model of the MSA. They do show also a lower insulin resistance, which is in line with the results we obtained with the adult mice, and we do obtain an engagement of the target by demonstrating that yes, this in utero administration of the molecular construct was capable of decreasing the GRK2 levels in these mice. So the take-home message is, the conclusion of this work is that GRK2 levels, they are upregulated into the brain of MSA patient by downregulating, decreasing the levels of GRK2 into the striatum or into the whole brain, protects, and I put it within bracket because we have to be very cautious, from developing nigrostriatal lesion and synuclein accumulation into the striatum, as well as insulin resistance. So GRK2, or the decrease in the levels of GRK2 might appear as a possible target. And also this was clearly a bench approach, so an approach limited to the lab, where we demonstrated that gene therapy might be useful. It is quite interesting to see that small molecules are worth trying with a CNS delivery, because while if you remember my introduction, where there was a demonstration that decreasing GRK2 levels might be interesting for obesity, for insulin resistance, for heart failure, and so on. Of course, the colleague researchers working in those areas have kept working, and they have developed small molecules that are now interfering with the levels or with the activity of GRK2, and they offer the possibility of using small molecules. The caveat, of course, is that those molecules were all developed for peripheral conditions, meaning they were not designed for entering into the brain. So if we were to try those molecules, we would have to do a lot of work in order to be sure that those molecules might penetrate the brain. But this is definitely a way to go, much more promising than gene therapy, which is clearly invasive. I would like to acknowledge, first of all, the support of the MSA coalition. This is extremely important for everyone, not only the money, of course, which is important for collecting the experience, but also the recognition that the project might be of interest. And here you have the name of the people who have been working on this project in the lab: Miguel Lopez Quina, Ana Delama, and they are both medical doctors, and So they are seeing patients, and they are also doing experimental work. Marie-Laure, Alzheimer, Marie-Hélène Coron at EduTech. And of course, Pierre-Olivier Famagu and Wassilios Meissner, my co-PI on this project. Thank you.
Thank you, Dr. Bezard. We appreciate all that you have just shared with us, and the exciting piece that there seems to be maybe some way that what we're looking at may make a change, a difference in MSA patients moving forward. We did want to have some questions, and unfortunately, since this is the first webinar and there was a small audience, we don't have any yet from the audience, but all of us who participated in putting together this did come up with some questions. Karina had several, and I know I have several. Karina, would you like to start with your question, one or two of your questions, to get us moving forward, to help us better understand, or maybe come up with some applications from what we've heard today?
Sure. I was actually curious about the gene therapy route that you just talked about, Dr. Bezard, and if the plan is to try to use, I'm guessing AAV9, because we know it crosses the blood-brain barrier, but instead of in utero delivering it, I guess systemically, or if there's other routes that are being looked at in the animal species.
Yes, so what we are using is a 2-9, so 9 basically, and AAV9 serotype, for exactly the reasons you are alluding to, which is a way for accessing to the brain without having to do a stereotactic surgery, so an invasive surgery. The research domain is full of promises, but they seldom realize into a practical solution. So yes, there was a lot of enthusiasm for the use of systemic administration of AAV9 for reaching the brain. But when we switch to monkeys, so to primates, which are closer to us, unfortunately, those hopes unravel to be false, because in monkeys it does not reach the brain. So even in monkeys, we have to go in utero. And in fact, it sounds a bit extreme, but in fact, most university hospitals, they do access to embryos almost on a routine basis, and therefore an in utero administration into an embryo is not an obstacle.
Okay, thank you.
I wanted to ask a question, Wassilios, I think it goes to you, but maybe both of you could answer it. You had a slide that showed that you saw some degradation, I believe, in the forebrain, and it was more active there than it was in the pons and the brainstem for that one particular discovery you made. Were you surprised that the forebrain would be impacted in an MSA patient, or at least in animals that are modified or that are modifying MSA or showing MSA?
I think, Erwan, you would need to clarify what you understand by forebrain. I think you mentioned it, that it includes the striatum.
Yes,
That's correct.
Yes,
it includes a striatum, so it's a gross definition of everything which includes the striatum and the many cortices. So it's difficult to be more accurate than that, because we had access to that kind of tissue, but it's not surprising. I mean, from the original paper published by Fares Basil and Wassilios Meissner as the last author, there was clearly an increase in insulin resistance in many places of the brain. So seeing this to be accompanied by an increase into the levels of GRK2 is not a surprise.
Okay. Karina, you have another one? Bouncing back and forth?
Yeah, I'm actually, just to follow up to the other one, I'm curious about the translation from rodents to NHPs or primates, if it's not translational to NHPs, if you think that would be an indicator of it not being translatable to the human population. I don't know if it's always necessary for it to work in the NHP before going into the human.
No, clearly. Anyway, and this is very sad for the researcher, but at the end of the day, especially for basic researcher like myself, we have to understand that the clinicians, they do not need to have preclinical data, basic science data, in order to develop a therapeutic. Of course, it is better to have knowledge and to have, let's say, an increase in confidence that yes, a strategy might be useful, but you do not need such level of information in order to launch a trial. Okay, so this is very important. So to follow up on your question, depending on the field, it's a question of habit. So in the field of Parkinson, for instance, you need absolutely to go through the primate before to reach the clinic. Mm-hmm. Because there has been fantastic models of let's say a copy of the symptoms in monkeys, not really of the pathology, but of symptoms. And therefore, if you want to develop a treatment aiming at alleviating the symptoms of Parkinson's disease, you would have to go through the monkeys. By analogy, most of the program include monkey experiment when dealing with neuroprotection or with compounds aiming at slowing down the progression of the disease. In MSA, it's very different. And the latest development of possible NHP models of MSA are one very recent. Professor Meissner and myself published two different models relatively recently, one using a delivery of synuclein using a viral vector into the oligodendrocytes, and one very, very recently using extract from the brain of MSA patient, where we extract the GCIs, so the inclusions contained into the oligodendrocytes, and we inject them back into the brain of monkeys, and they develop some pathology over time. It's a very slow process. But these models, they are interesting, but they are exploratory, and in no way they are yet implemented into the translation process of science. So today, if you demonstrate things into the few MSA mouse models in the States, there is one or two in the States, one in Europe, and also in Japan. It's enough, considered to be enough for moving to the clinic. Maybe Professor Meissner wants to add something to this.
I fully agree with you that preclinical data are there to make us confident that there is some biological target engagement. This is really important. But I would really like to stress that beyond the classical preclinical models, what we have been doing is to include post-mortem tissue of patients, because if you don't see any of these changes in patient brain tissue, then I'm not sure that what we see in the preclinical models is really meaningful. So this is very important, and the field has really moved forward, and especially in another topic, which is the alpha-synuclein topic, and I'm sure that you will have also some webinars about alpha-synuclein and also the potential treatment target, that here the patient tissue is really fundamental to better understand differences in alpha-synuclein species between, for example, MSA patients and patients with Parkinson's disease. And there have been very beautiful papers in the last 2 years that really helped us move forward in the understanding. And these data are really fundamental and very, very important as well.
Is this going to work for both of the, well, I guess for any of the different hybridizations of MSA, MSAP, MSAC, and the ones that are atypical Parkinsonism? Is this something that's more focused for MSAP, or do you see this being beneficial for any of the three different, I guess, versions of MSA we're looking at now?
So to be very honest, we have looked in our paper. I think it was only MSAP, okay. But there is, of course, I have no proof, and we would need to look into it. But I cannot see any reason why there should be a difference between MSAP and MSAC. Maybe the regions would be different. So this comes back to your question, Larry, about stratum and pons and cerebellum, and potentially in patients with MSAC, you would expect that these changes are more overt in the brainstem and the cerebellum. Yeah, we would need to look at this, but I don't really see why there should be differences.
Okay. So what would a clinician, we're going from bench to bedside, what would we have as a clinician see when the clinician is observing these patients that are coming in, like, for example, the randomized sample trial you're doing now, or those that are in University College London? what are the clinicians going to be seeing as differences if this works?
Yeah, so currently, I mentioned that the endpoint that was selected in that study is a scale, a clinical scale that allows us to assess, or this is one part, which is I would say, a patient questionnaire where the patient is asked questions about activities of daily living, and depending on the severity on impairment, that allows to sum up a score. And then there's a second part, which is basically a neurological exam done by the neurologist that will also allow to give a score of the overall motor disability. And this is how we do today. And I mean, you know very well, because there is this scale that has been developed 20 years ago, and it has really helped us in natural history stories. It has helped us as primary endpoint in clinical trials. And this is how we're doing it today. But of course, we don't, in that particular trial, if you're just using the clinical outcome, we don't really have a biological measure, which would be interesting. But I can tell you that I'm sure that the clinical trial will also look a little bit at these exosomes I mentioned, these small vesicles in the blood you can detect, and that confers some information from the brain. And we have some experiences from the field in Parkinson's disease, because there have been a few clinical trials, and the colleagues in London, Tom Fultini, he has conducted a trial in Parkinson's disease patients that was published in 2017. And beyond the fact that they observed a small but significant signal on the progression of motor symptoms, they also assessed, in collaboration with Dimitris Kapagnianis from the NIH, these exosomes, and found differences between patients treated with the anti-diabetic and those who were not. And we have completed a trial also in Parkinson's disease. So we're a little bit more in advance in Parkinson's disease compared to MSA, in 150 patients with Parkinson's disease in France, where half received one of these anti-diabetics and the half placebo. And we will also look into these biological markers to understand if there is an effect, if that translates into some biological signal, because I think in the future, and this is really the main theme for the clinical trials in the future, beyond the clinical assessment and the understanding how a clinical trial and a drug makes the difference to a patient, because this is at the end of the day for the patient the most important. Right. But I think it is important also to understand what are potential effects on the biology, on the biological target, and also, for example, on other surrogates such as imaging. We have seen really over the last 10, 15 years, a huge progress in our understanding how the disease progresses in the different brain areas, and we can measure it. And it has been used as what we call a secondary outcome. So something secondary outcome, you mean, or exploratory outcome, you mean something we would like still to learn a bit how this is doing over time. And we have seen recent trials that have used imaging, and I'm very, very confident that over the next few years, we will increase the way to measure really the impact, not only on the clinical side, but also on the biological side. And that's important.
Okay. Well, we were going to, before we moved on, we were going to ask what are any future trends you see in this area? And I think you've just addressed several of them with what you just discussed. I did hear you were thinking, or you had done one where you thought you would be able to do the whole brain. Is that something that would be a future trend someday in the future, hoping to be able to treat the whole brain of an MSA patient, or are we still looking at areas as we move forward in that space?
Yeah, I think it's really very, very, very important to get that whole approach, especially in disorders where really many regions are suffering. And this is when it comes, I hear again, the Parkinson's disease field is more advanced, and there have been some approaches, gene therapy, very focused with injection, for example, on the putamen, or also grafts injected in one region. And I'm really afraid that would not be enough. So when I'm advising companies, sometimes I always say to them, look, you would need to have a whole brain approach, because if you're just focusing on, for example, the substantia nigra, and just for the patients and the caregivers who are connected, this is the region where the dopamine neurons are. If you are just focusing on these dopamine neurons, that would probably not be enough to cover really the broad area of symptoms patient may have.
Okay. But luckily, I mean, research evolves in parallel. So we have colleagues working very, very actively towards the development of new vectors capable of translating the whole brain. By translating, I mean bringing the genetic correction, the gene correction to the whole brain. And this is done in the US, in Europe, in Japan. So we do have a huge effort from the industry, which is good because it means that there is money to achieve this goal. And I am very confident that sooner or later, those tools will become available.
Okay, thank you. Thank you very much for what you've done. Karina, I guess you're going to be closing it out.
Oh yeah, sorry, I just had one other question.
Oh, you go right ahead.
Okay. I was just curious about insulin resistance as a whole, because you start seeing practitioners now using it as a marker for aging, and obviously these age-related diseases, you mentioned Parkinson's and Alzheimer's, and the work that you focused on here was specific to MSA, but I'm just wondering if neurons or oligodendrocytes have been shown to have insulin resistance as well, like in Parkinson's or Alzheimer's disease, if researchers have already looked at those, since you mentioned they were a little bit further along potentially.
Yeah, I mean the data is really growing and really strongly growing. So from the Alzheimer's fields, it's over the last 15 years that there have been really a lot of information in line with insulin resistance. What is interesting here, when you look, for example, at these exosomal markers I mentioned, the insulin resistance markers in the blood of patients, and we're looking at a cohort of diabetes patients, then of course there is an increase of peripheral insulin resistance and also on the brain as well. But when you look then at the cohort of patients with Alzheimer's disease, and you know, I mean, this is always somehow the advantage of a larger disease. You're putting together a cohort of healthy elderly patients, and then you follow them for 10, 15, 20 years, and they come back to the office and have an examination every year and have blood sampling and imaging. And then once they declare disease after 15 years, you can go back. You can go back to the first samples and you can have a look what happened at that time. And what is really fascinating is that in patients with a diagnosis of Alzheimer's disease at a certain point in these cohorts, when you go back 10 years, they already had insulin resistance in the blood. So this brain marker I was mentioning. So this is really a very, very strong signal, which has led somehow also in the literature among the colleagues to the term of type 3 diabetes, because we have now two types of diabetes. The first is rather in the younger patients who lose the capacity to produce insulin because of an autoimmune disease, and then we have the type 2 diabetes, which is rather the phenomenon in the elderly population. And so this term is coined type 3 diabetes in Alzheimer's disease. And Parkinson's disease, the data from these large cohorts are less evident, but it's really going also into this direction. What we know now is if you have diabetes and Parkinson's disease, then you're really doing worse in a way that the disease is progressing much more rapidly than would in a patient without diabetes. And we're currently challenging this concept also in our preclinical mouse model, because we have exposed these MSA mice to a diet that induces diabetes to see if we could boost the process in the brain. So this is the PhD work done by Anna Delamar, who was also mentioned on the final slide from Erwan. So we are trying to gather more information to make this story a little bit more robust and to understand more the mechanisms behind. Yeah,
makes sense. Thank you. Well, with that, we want to thank you both, Dr. Meissner and Dr. Bezard, for taking the time to share your research today. We also want to thank each and everyone who donated monies to the MSA Coalition in the past and to help fund these two projects. This year's call for proposals has just gone out, so we'll soon see our 9th year of pre-proposals, and by the time of the MSA Coalition's annual conference, we should have another group of researchers ready to start research into their hypotheses. So from bench to bedside, this is the promise and hope of our MSA research. Thank you all for joining today.
Thank you. Thank you, Dr. Bezard. Wassilios.
Bye-bye.
Bye. Have a good day.
Yes, you too.
Bye-bye.
Thank you, Casey, for helping. Appreciate it.